Okay, chapter 12, chapter 20, production. So we're going to look at primary production, photosynthesis, and chemosynthesis as ways of generating energy that supports everything. So the energy in ecosystems originates with primary production by autotrophs. Net primary production is constrained by both physical and biotic environmental factors. Global patterns of net primary production reflect climate constraints and biome types. And secondary production is generated through the consumption of organic matter by heterotrophs. The deep sea was once thought to have few forms of life because of darkness, and so no photosynthesis and tremendous pressure. And so it's assumed that because there's no light, you can't have primary productivity, so there must not be a lot of life. But in 1977, we discovered a new type of undersea or deep sea community supported by hydrothermal vents. And this is an image of a black smoker, which is a hydrothermal vent discharging superheated water that can be as high as 400 degrees C. It's rich in iron sulfate, and there's a lot of iron sulfide. There's a lot of sulfate-reducing bacteria that often live in symbiosis with other organisms. There's the, is it Riff? It's in the next slide. Riftis or Riffia, the tube worm. I should put a picture in of it. One of my good friends, Andrew, discovered a species of Yeti crab off Costa Rica. And so these are crabs that they're white and they have a bunch of hair on their arms, and they kind of do this like dance where they wave their arms around. And on their hair of their arms are these sulfate reducing bacteria that are using sulfide as an energy source, and then the crabs then eat the bacteria growing on their hairs. It's kind of cool. Well, actually, here these are squat lobsters. These are different than the Yeti crabs. But you can see mussels and shrimp. And so they're all feeding off of these some way of the energy source of chemosynthesis from this hydrothermal vent. Riftia is the tube worm. They're really pretty. They have like a big long red plume, and you'll find a whole hydrothermal vent associated ecosystem with clams, shrimp, crabs, polychaetes, etc. And really high density of organisms and a really high diversity in biomass relative to the other areas. And actually I hadn't thought about this, but this would be a worthy question, which is looking at the distribution of hydrothermal vent organisms, like what would you expect? And presumably they would be a patchy distribution because their food source is very patchy. And so they're not evenly distributed amongst the seafloor, right? They're associated with this food source, hydrothermal vents, and iron sulfide with sulfate reducing bacteria, or a lot of archaea that consume methane as well. But nonetheless, it's a patchy distribution because it's associated with this food source. So the distribution of Riftia would not be even amongst the seafloor. It would be patchy because it's associated with hydrothermal vents. Would be something I'm going to probably write down. So the distribution of Riftia along the seafloor. So where these organisms get energy, photosynthesis is impossible, and the rate of which dead organisms from the upper ocean accumulates on the bottom is very low. In addition, water coming out of these vents is extremely hot and full of minerals that would be toxic to most organisms. So these communities are surviving via that chemosynthetic pathway. This is kind of an interesting diagram because, like, two things. One is it was like hard to get published, and sort of maybe a good aspect of what my advisor would call the Matthew effect, which is more famous authors get their work published more easily than non-famous authors. In this instance, the Lindman's advisor, I forget who it was, but was famous enough where he could kind of write the editors and say, no, you need to publish this. And they're like, okay. But here it's looking at energy transfer through a lake ecosystem and having sort of interesting, like these two different pathways, but this connection with ooze or bacterial ooze, where you can have solar radiation come in through the planktonic system of going to plankters, zooplankters, and plankton predators, and then moving on to swimming predators, fish, etc. Or you can have it go through this pondweed mechanism where it's going through either attached algae or floating algae, then onto browsers, benthic predators, and then into swimming predators. But there's this loop in here with both dissolved nutrients and then taking into account the bacterial loop here as well. And so here, instead of putting taxonomic categories, what Linneman really did is group them into functional categories based on how energy was obtained. And so it's kind of zooming out. If we want to understand how an ecosystem works, following the energy, so that you often hear like, follow the money to find corruption or whatever it is. Well, here it's following the energy. And so if we want to understand how a system works, we need to follow the energy through the ecosystem. And so this was groundbreaking in that, we're really saying that, which is true, what we're really saying is to understand the foundation of how ecosystems are structured, we need to be able to follow the energy more so than the individual players within that ecosystem. And there we can derive patterns that are more universal because it's not dependent on a single organism. It's dependent on the broad function of them. These ones are fixing carbon, and there's these different exchanges. And so looking at that way, we can start to build I would argue a more universal understanding of how energy flows through ecosystems and therefore how they're structured. So I was kind of surprised because the term ecosystem is relatively new. I guess, I don't know if 35 is new, but it feels relatively new when you go back to Darwin, etc. So the term ecosystem was first used by Tansley in 1935 to refer to all the components of the ecological system, biotic and abiotic, and the influence of flow of energy in the elements. The ecosystem concept integrates ecology in other disciplines, geochemistry, hydrology, atmospheric science, etc. So this is the big move, is moving from looking at communities of organisms to looking at the ecosystem, which then includes the environment, the abiotic environment as well, which makes sense, right? Like, the type of climate, the amount of sun, the type of local geology, all of those things impact the ecosystem. Oops, sorry, I didn't say I jumped. Primary production is the chemical energy generated by autotrophs during photosynthesis and chemosynthesis. It's a source of energy for all organisms, from bacteria to humans. You could argue archaea to humans. All accounts for the largest movement of carbon dioxide between the Earth and the atmosphere. Fossil fuels are derived from primary productivity. I'm not sure if it's here or later. So one interesting thing about this, how we derive energy from fossil fuels, this is energy, this is carbon that was fixed by photosynthesizers, and within that compound there's energy. And so when we break that compound, we get energy, and carbon dioxide is released. There's a lot of talk around carbon recapture as a way of mitigating climate change. One of the issues is the release of carbon generates a whole lot of energy. And so by reverse, to capture carbon and bring it back into a different form requires a lot of energy. And so it's one of those things where really a lot of our carbon capture technologies will use more energy then so let me there's another way to think about that. So if we were burning coal to power a carbon capture device, we would have to release more carbon via coal burning to power the carbon capture device than carbon we would be capturing. And so at a certain point, it's not energy efficient, where you can say, well, what if we powered carbon capture devices by wind or solar, etc. Sure, but wouldn't it be? It would probably reduce the overall carbon in the atmosphere by more if we use that wind and solar power to replace fossil fuel burning sources of energy, versus using that solar or wind energy to try to capture carbon in the environment. We'd actually be better off by switching from fossil fuel burning to green sources of energy. So you have to think about, like, there's a lot of energy stored in these compounds from carbon fixation. And when we break those bonds, release that carbon dioxide, we're generating a ton of energy, and to put the genie back in the bottle takes a lot. Kind of an aside, but still worth thinking about. Energy assimilation by autotrophs is stored in carbon compounds in plants. This carbon is the currency used to measure primary productivity. Primary productivity is the rate of primary production. Rate isn't important there. So primary productivity is a rate, meaning amount of carbon per something. And it's usually per area per time. So grams carbon fixed per meter squared per year, per day, per hour, some sort of per, because it's a rate. Gross primary productivity, GPP, is the total amount of carbon fixed by autotrophs. Gross primary productivity is controlled by climate through the influence of photosynthetic rate. And leaf area index, LAI, leaf area per unit of ground. We'll talk a little bit more about the leaf area index. But gross primary productivity is the total amount of carbon fixed by autotrophs. We'll get into gross and net. I'll give an example to differentiate those two if you're not an accountant. Leaf area index varies by biomes. Less than 0.1 in the Arctic tundra, which means less than 10% of the ground surface is leaf cover. In boreal and tropical forests, they can have an average leaf area index of 12, so greater than 1, meaning for a meter squared, if you were to start at the ground and go up, if you were just like think about a point on the ground going up, you would pass through 12 different leaves before you got to the air, to the atmosphere above the leaf cover. Conversely, you could probably think about it. It might make more sense is if you're a beam of light, you would hit 12 different leaves before you go down to the ground. So there's these overlapping layers of leaves, which makes sense. Like you walk in a forest and there's the understory, the overstory. Trees don't just have one layer of leaves; they often have many different layers of leaves, right? So that's what we're looking at here. And this is the diminishing returns for added leaf layers. And so this is the net, although we haven't told you what net means yet. Well, you'll figure it out. The net carbon uptake by leaf layer. And so here we can look at the number of leaf layers or leaf index, but the increase is smaller for each additional layer. So here if we go from 0 to 1 layer, we have a fair amount of increase of the net carbon uptake, grams per meter squared per hour. So remember, primary productivity is the rate of primary production, rate. So it's grams per hour, and we want to normalize that by some unit of area. So grams per meter squared per hour. And then we add a second layer, we do get an overall increase in net primary productivity. There's more carbon being fixed, but it's marginally less as we go up to each layer. Up to layer 15, where we add an extra layer and we're capturing a little bit more sun energy, but not a lot. And this will go until basically gross primary productivity, net primary productivity would go to 0, meaning the additional layer would not have any additional benefit beyond the gross, or rather, plant respiration equals gross respiration is probably another way of thinking about it. There's no additional benefit. You can't make a living. There's no extra carbon left over for net primary productivity. Plants use about half the carbon fixed in photosynthesis for cellular respiration. So plants with a lot of non-photosynthetic tissue, e.g. trees, have a higher respiratory carbon losses. Respiratory rate increases with temperature, so tropical forests have a higher respiratory losses. What we're getting at is the difference between gross and net primary productivity. And so plants are true. It's true that they are taking in carbon and releasing oxygen, but they are also requiring oxygen themselves and releasing carbon dioxide. They are living organisms, so they're respiring. So net primary productivity is the amount of carbon left over after respiration. So you can think of this in like a retail store. If you sold a shirt for $20, that would be your gross profit minus paying $10 for the shirt and everything else, then you would net $10. So net is the amount left after you pay your bills, and in this course, in case your bills is respiration. So net primary productivity is the amount of energy captured by autotrophs that results in an increase in biomass. So this fixed carbon has to go somewhere. They don't just put it in Bitcoin. It's in a physical medium of fixed carbon, so it contributes to biomass. It can be stored in a variety of different ways, but it's still measured in biomass. It's still there somehow. It exists in a physical state. So net primary productivity is the energy left over for plant growth and consumption by detritivores and herbivores, etc. Plants can allocate carbon to different tissues. And so, let's jump back and let me go back to this again. So if for every gram of carbon fixed, the plant uses about half of that gram to serve for cellular respiration, and then that varies between the ratio of overall biomass to photosynthetic area, meaning that if you're huge, if you're a tree with a giant trunk, there's a lot of biomass there that is respiring, right? That's using oxygen, releasing carbon dioxide, and you have a relatively small amount of photosynthetic area for your mass, compared to like a grass that, it's long, skinny, and thin, and most of its mass is photosynthetic. So you would have a different ratio of a different percentage that can be captured in net primary productivity. So for most organisms, net primary productivity is about 50% of gross primary productivity, but for a tree it might be lower, like 25% or so. As well as tropical forests. So if we had a grass in a temperate area and a grass in a tropical area, the grass in the tropical area would have a lower percentage of net primary productivity compared to a grass in a temperate area where it's cooler. They would have a higher percentage of net primary productivity relative to gross primary productivity. So plants can allocate carbon to different tissues. Grassland and desert plants allocate more net primary productivity to its roots because soil, nutrients, and water are scarce. And we'll look at we'll look at that next. And so net primary productivity can be allocated to storage products, i.e. starch. And up to 20% of net primary productivity can be allocated to defensive secondary compounds, toxins, things that make you taste bad, to herbivores. So here is a figure looking at percentage of net primary productivity allocated to roots. Tropical forests, a relatively small amount of net primary productivity is allocated to the roots. Temperate grasslands, very high amount. So there's two different things going on here. One is competition for light. If there's increased competition for light, you would put less of your energy into roots and more into growing photosynthetic biomass, either larger leaves growing up higher to shade out your neighbors. So if we look at areas where there's not a lot of competition or there is a lot of competition for light, you would assume that you put less energy into your roots, as well as areas where you need to conserve water, nutrients, or other energies. So here you need to conserve a lot of water, so you're going to put more energy into your root biomass, where you have basically a store of energy, as opposed to building out more photosynthetic biomass, because photosynthetic biomass, when you produce more area, you're now going to lose more water through transpiration. So there's an advantage to, can I build a photosynthetic structure that's efficient and it will generate more energy than I need, and I can store some of that somewhere else and be able to have that in the most efficient manner, versus can I just expand and put out a massive amount of photosynthetic biomass where, sure, I'll generate more energy, but then I'm become water limited. Something like temperate grasslands or tundra, short growing seasons, I need to capture as much energy as possible, but I need to be able to store that to get through a long winter. Someplace where there's an annual above ground growth, where I want to store most of the energy below ground. So I have an above ground vegetative biomass that grows in the spring, photosynthesizes like crazy, maybe dies off in the fall. And I've got enough energy stored in my my root biomass where I can then regenerate that photosynthetic capacity in the spring. So generally, you can look at this distribution and explain it by either competition for light, needing to store energy to get through seasonal cycles, or needing to conserve water or nutrients. Net primary productivity varies during succession as species composition, leaf area index, and ratio of photosynthetic to non-photosynthetic tissues change. The highest net primary productivity is usually during intermediate stages when plant diversity and nutrient supply tends to be the highest. Now, this is pretty interesting. Yeah, I'll go to the next one, and then I'll jump into my idea. So this is meaning, like, we look at the succession of forest or of plant ecosystems. Generally, there's this intermediate stage where you've got some of the early succession and you've got some of the late succession. You've got a broad diversity of plants, and there generally is the highest amount of net primary productivity. Later successions where you're could be dominated by, for example, one large tree. That tree's shading out all of the other understory plants, and generally you can have a decrease in net primary productivity. So in this case, our final stage of succession isn't necessarily the most productive. And so old growth forests, net primary productivity may decrease as leaf area index and photosynthetic rates decrease. But old growth ecosystems have large pools of stored carbon and nutrients that provide habitat for many animal species. So a decrease in net primary productivity over time is less pronounced in grasslands than forests. Let me look at this example here quickly. So I'm going to go back to this resilience idea. And we can look at two different ecosystems. And so this is one area in two different ecosystem states. And so we can have the Gary Oak dominated ecosystem as an intermediate station. Here, one second. So in the system, we can think of a Gary Oak dominated ecosystem as an intermediate stage, and then a Doug Fir dominated system as the climax stage. And we can use these two to think about both how that impacts net primary productivity, where we can have, in theory, increased net primary productivity in the intermediate stage, where we've got a mix of Gary Oaks, a lot of understory, in this case, things like chocolate lily, camas, other lower understory plants, where when we go to a Doug Fir dominated system, we've got primarily Doug Firs with a shaded understory that's receiving less light. And so in this case, the intermediate stage can actually be more productive. And if we think about these as different stable states, the social pressure here is keeping Gary Oaks in the intermediate stage and preventing the ecosystem from moving to the climax stage of a Doug Fir dominated system. So in this case, social pressure, i.e. in this example, burning on a rate of about 4 to 7 years is how often the Pacific Northwest was typically burned by low light fires, keeps Doug Firs from dominating and becoming the climax stage of a Doug Fir dominated ecosystem. And that social pressure, i.e. fires lit every 4 to 7 years, keeps the ecosystem in this Gary Oak stable state. So one example of how to think about an intermediate stage versus a climax stage and how that relates to primary productivity. So how do we measure this stuff? Turns out there's a bunch of different ways of measuring net primary productivity, and net primary productivity is the ultimate source of energy for all organisms in the ecosystem. Well, yeah, except for the photosynthesizer itself, right? They're getting their energy via the gross primary productivity, but net primary productivity is the amount that's left over. That's the amount of carbon fixed that is available in biomass for things to eat. Variation in net primary productivity is a metric for tracking ecosystem health, and net primary productivity is associated with the global carbon cycle. In terrestrial ecosystems, we can measure increase in plant biomass during the growing season by harvesting the plant tissues in experimental plots. So in your lawn, you can have a plot, cut off all the grass, weigh it, throw your quad somewhere else. Later in the season, cut down all the grass, weigh it. Okay, the difference between those two is the amount of net primary productivity that happened between those two sampling intervals. And this gives a reasonable estimate of above ground net primary productivity if corrections are made for herbivory and mortality. Meaning if some grasshoppers come by and chew on your grass, you need to account for that as well. That not all net primary productivity stays within the plant forever, because there's herbivores in the world and they consume that plant matter. Measuring below ground net primary productivity is difficult. Fine roots turn over quickly as roots grow and die. Roots may also exude carbon into the soil, transfer it to our friends the mycorrhizal fungi. And if you harvest must be frequent and additional corrections are needed. So you have to be able to dig up and get all of the root biomass when you dig up the plant, which can be hard because there are all these fine roots going off in different directions that break, as well as, how do you differentiate roots from this one plant you're trying to measure versus other roots that are overlapping and all intertwined. It can be a bit of a mess. So they invented this this device. It's a mini Rhizotron, which I think is kind of a funny word. Mini, small. Rhizo, so like mycorrhizal fungi, and Tron, I assume just like techie sounds cool. The Tronomatic. I don't know if that's how they came up with the name, but there you go. It's basically a camera. It's basically a clear tube that they can put in there with a camera, and so then they can run the camera down and measure the amount of root biomass growing around that clear tube, which allows you to make repeated measurements through time without having to dig up and kill everything. And so there's an advantage there. Harvesting techniques are impractical for large or biologically diverse ecosystems. So if you're trying to measure the below ground biomass of an onion growing in your garden, you can plant an onion and have it separated from everything else. So when you dig it up, you're ensuring you're going to get all of that biomass. But now imagine walking into, a natural ecosystem where there's just a whole bunch of plants piled on top of each other. How do you separate whose roots are whose? It gets quite difficult. We can also use chlorophyll concentrations as a proxy for gross primary productivity and net primary productivity. So chlorophyll is, the function, serves the function of fixing carbon. The amount of chlorophyll and primary productivity vary through time and by species, but if we work that out, then we can start to use chlorophyll as a measure for primary productivity. The advantage of chlorophyll is we can measure it with satellites, drones, low-flying aircraft, a wide variety of things. And what we're doing is we're measuring the spectral signature here of measuring wavelengths of light and the reflectance. And so here is one for vegetation, and we can see vegetation reflects a lot of light in the near infrared spectrum here, between 700 and about 1400, as well as there's depressed light in the blue and red visible spectrums because that light is being absorbed by the plants, and so it's not reflecting that light. They reflect green, which is why they look green. They're absorbing red and blue wavelengths here. So the difference in reflectance here tells us that there's vegetation because it's absorbing red and blue, reflecting green. Plants are green because they're reflecting green light, not because they're absorbing green light. And so we can use the ratio of this depression to the amount of the near infrared reflectance as a proxy for chlorophyll. I'm not really, this is interesting to know. I'm not going to say here are some values and calculate the NDVI. If you want to learn more about this, take Remote Sensing by Wallen. This is more what he does. So the NDVI is Normalized Difference Vegetation Index. And so it's a way of comparing effectively photosynthetic capacity via chlorophyll. And it's the ratio of near-infrared wavelengths minus the amount of red divided by the near-infrared radiation plus red. And so it's using both of these components of a lot of near-infrared radiation and then absorbing red to come up with the relative index of vegetation. So this is useful particularly if you're going to follow a system through time, or maybe compared within an ecotype within a certain area. If you have early and late successional areas, you can compare the two and look at how the NDVI varies. Vegetation has a high NDVI value and water and soil have low. NDVI over large spatial scales is measured using satellites. We can now attach these to drones, and actually it's used a ton in agriculture. A lot of this technology is driven by needing to monitor farms that are on the order of tens of thousands of acres, and so they fly not with drones, actually they're fixed wing, but they're drones, they're not quadcopters. And they fly back and forth and measure infrared light, and then based on that they can say, this little patch of corn isn't doing as well. Remote sensing can be used to estimate carbon dioxide uptake and net primary productivity, deforestation, desertification, atmospheric pollution, etc. Remote sensing allows net primary productivity to be measured frequently at spatial scales up to the entire globe. So that leads to maps like this where we can look at what does net primary productivity look like around the globe. And you can see hot spots in the ocean for upwelling areas, which makes sense. and generally where our most of our primary productivity is occurring. Net primary productivity can be estimated from gross primary productivity and respiration methods, meaning if we know how much carbon is fixed and we know the respiration rate, we can figure out net. So change in carbon dioxide concentrations is measured in a closed chamber. Inside a tented chamber, carbon dioxide is added by the respiration of plants and heterotrophs, including soil organisms, and uptake of carbon dioxide is via photosynthesis. The net change in CO2 is the gross primary productivity minus total respiration. This is the net ecosystem exchange. So what this is taking into account are heterotroph respiration as well. So if we're looking at the overall net ecosystem contribution of really, how much carbon dioxide is fixed and how much oxygen is being exported from that environment, we're taking into account heterotrophs, soil microbes, insects. We'll look at one that includes a large herbivore as well. So heterotrophic respiration must be subtracted from net ecosystem exchange to get net primary productivity. Thus, net ecosystem exchange is more refined estimate of ecosystem carbon storage than net primary productivity. So what this is meaning is we often, if you have a plant and it fixes, let's say a kilogram of carbon, then it utilizes half of that kilogram for respiration. So it's releasing that back in the environment. And then half of that plant gets eaten and then respired. The amount of carbon storage is now the leftover of that, which would be a quarter in that example. So we start off with a kilogram of carbon fixed. The plant uses 500 grams for respiration. Now there's 500 grams of net primary productivity. And then the heterotrophs consume half of that 500 grams, 250 grams, and respire that. Now the plant has 250 grams left as carbon storage. And so we can look at the net ecosystem exchange, which is equal to the gross primary productivity minus AR, which is the autotroph respiration, how much the plant's respiring, plus the heterotroph respiration. So in that example I gave, our gross primary productivity would be 1,000 grams. The autotroph respiration would be 500 grams, and the heterotroph respiration would be 250 grams, leading to a net ecosystem exchange of 250 grams of carbon dioxide. It's worth knowing that, and it's worth thinking about what if in this system of forest and grasslands, what if an invasive came in that took over the entire area, but wasn't consumable by the herbivore? How would that alter this equation, given some estimates of its gross primary productivity and its respiration rates, autotroph respiration? So think about that and the role of heterotrophs in the system. So this is kind of an interesting one, and this is done a lot in experimental forests. The Harvard Experimental Forest and a few others where they've got a whole bunch of infrastructure in a small area. They'll look at eddy correlation or eddy covariance, where net ecosystem exchange is estimated by measuring carbon dioxide at various heights in the plant canopy. So the idea is that we can measure all these individual components and try to put them all together in a model, or we can just measure how carbon dioxide is changing within this little patch of forest. The gradient of carbon dioxide develops because of photosynthesis and respiration. During the day, CO2 decreases in the canopy with photosynthesis, and at night, CO2 is released is higher in the canopy. So again, we often think about plants are absorbing carbon dioxide, which is true during the day, but at night they're still respiring, as are all of the heterotrophs in the environment. So you could have big swings in carbon dioxide in a well-insulated canopy, meaning that during the day carbon dioxide is being drawn down, and in the evening it goes back up. So you do this by putting up these complex towers with a bunch of different sensors. Here we're looking at height from 0 to 20 meters, and then hours. Presumably this is done where this is noon and then this is in the evening. This is concentration of carbon dioxide, which should be on this axis, which is not really, but so there's a kind of a topographic map here of increased carbon dioxide between midnight and 6 a.m. and increased carbon dioxide. 20 hours would be 8 p.m. And so here during the day it's less carbon dioxide. In the evenings at night it's increased carbon dioxide. So that would be measuring using eddy covariance to estimate net ecosystem exchange. So the difference between those would tell you that. And how much more do I got here? 2 more, okay. In aquatic habitats, it's a lot different. Water is different than land. In aquatic habitats, phytoplankton have a short lifespan, so biomass at any given time is low, and harvest techniques are not used. Photosynthesis and respiration are measured in water samples, incubated on-site in light and dark, and differences of these rates equals net primary productivity. What does that mean? I will show you. Here it is. So, let's get this a little bigger here. So here is a very classic way of doing this. So what you do is, if you want to figure out what's the primary productivity of a ocean ecosystem, freshwater, etc., you go out and say your photosynthetic max is at 10 meters. I mean, 10 meters water depth, there's the most amount of chlorophyll. You send, it's called a Niskin bottle. It's effectively a bottle on a string down to 10 meters. You push a button, it closes this bottle, it brings it back up. Now you have a sample of water from 10 meters. You take some of that water, you put it in a bottle that you cover in electrical tape, and you put some of it in a clear bottle. Clip both of those to a line, drop them down, measure the amount of oxygen in the bottle, and send them down to depth, leave them there for a day or two, pull them back up. The light bottle is telling you the amount of oxygen produced by photosynthesis minus the amount of oxygen consumed by photosynthesis, your net primary productivity. Meaning this is a closed system here, so if we start off with the value of, say, 5 milligrams of oxygen per liter, we put it down for a day, pull it back up, it has 10 milligrams of oxygen per liter. That was an increase of 5 milligrams per liter per day. So our net primary productivity in that case would be 5 milligrams of oxygen per liter per day. The dark bottle is telling us the amount that's consumed by the plankton. So this is your respiration. So you've got the same initial conditions. You put your water in here with phytoplankton in it. You measure the amount of oxygen at 5 milligrams per liter. You put that down. You let it sit for a day. You pull it back up. Now it has an oxygen value of 2.5 milligrams per liter. So we've lost 2.5 milligrams of oxygen per liter in a day. So our respiration would be 2.5 milligrams of oxygen per liter per day. So that's our respiration and our net primary productivity. If we want gross, so net's after expenses and gross is total productivity. So if we want gross, we would take the 5 here, our net primary productivity of 5, and then add the amount that we lost to respiration, should be 2.5. So our total production, our gross primary productivity would be 7.5 milligrams per liter. You can also do it by the simple difference between the two. So if we recall, after 24 hours, the light bottle had an oxygen value of 10 milligrams per liter. The dark bottle had a measurement of 2.5 milligrams per liter. They both have the initial same starting condition of 5 milligrams per liter. So 10 minus 2.5 is 7.5. So it gets you the same answer of gross primary productivity being the difference between the light and the dark bottle. So that's how we measure primary productivity in the ocean. So it's a lot easier than a forest ecosystem where you've got above and below and all this kind of stuff. We can also, wrong button, we don't want that. We want to go back to our last slide and then take a break before the next section. We can also use chlorophyll via satellites, which works very well and is used across, obviously wide swaths of the ocean. It's pretty interesting because it only measures like the top centimeter or two and then extrapolates out the water column chlorophyll value. But it's really good for getting like blooms, broad relationships between upwelling and primary productivity, etc. Okay, we'll stop here and we'll come back for the next couple sections.